Today we are going to study 1.2 carbohydrate, which we aim students able to state the classes of carbohydrates, identify the structure of alpha glucose and beta glucose, which are monosaccharide, maltose, the example of disaccharide, and cellulose, the example of polysaccharide. Third, describe condensation and hydrolysis processes. And lastly, illustrate the formation and breakdown of maltose. The classification of carbohydrates can be done based on the number of sugar units. If the carbohydrate consists of only one unit of sugar, it's called as monosaccharide, which is also simple sugars. If the carbohydrate consists of 2 units of sugar, it's called as disaccharide, which is also double sugars. Di means 2. And if the carbohydrate consists of more than 2 sugar units, usually they consist of large number of the sugars, it's called as polysaccharide, which is also known as complex sugar. There are 3 examples of monosaccharides, which are glucose, fructose, and galactose. Each of these can be found in your food source. Monosaccharide is the simple sugar which consists of only one unit of sugar. And the term carbohydrate already implies the meaning behind the structure of the monosaccharide, whereas carb represent carbon and hydrates represent H2O. So the simplest formula or empirical formula of monosaccharide can be represented by CH2O in bracket N, because the N represents the number of carbon can be found in one monosaccharide. The classification of monosaccharide can be divided according to the location of function group and the number of carbons. The monosaccharide with the smallest numbers of carbons are trioses, means that this monosaccharide consists of 3 carbons, such as glyceraldehyde and dihydroxyacetone. And we have the monosaccharide consists of 5 carbons, which are pentoses. And lastly, the monosaccharide with the 6 carbon structure called as exoses. However, the monosaccharide can be further divided according to the function group. For example, trioses. If the function group C double bond O or carbonyl group located at the C1, the glyceraldehyde is also known as aldoses. And if we refer to dihydroxyacetone, even though they have the same chemical formula as the glyceraldehyde, which are C3H6O3, but because the function group located at the C number 2, so dihydroxyacetone is also known as ketoses. In our syllabus, we are going to focus on the monosaccharide that consists of 6 carbon with the formula C6H12O6. There are 3 common examples of monosaccharide with the 6 carbon, such as glucose, galactose, and fructose. But because of the different location of the function group, C double bond O, glucose, and galactose are grouped as aldoses, and fructose is grouped in ketose. And glucose is the example of monosaccharide, which is also the hexose, because the structure consists of 6 carbon, which is folded in a ring structure, not a straight chain. Same goes to the beta glucose. Another similarity between alpha and beta glucose is if we focus on the hydroxyl group of carbon number 2, 3, 4, they show the orientation below the plane of the ring structure, above the plane of the ring structure, and below the plane of ring structure, or we say BAB. Same goes to the beta-glucose. The location of the hydroxyl group from the carbon 2 until 4 is BAB. However, the main difference to name it as alpha-glucose and beta-glucose is actually the location of hydroxyl group at carbon 1. Alpha-glucose, the hydroxyl group is located below the plane of ring structure, but for beta-glucose, the hydroxyl group is located above the plane of ring structure of carbon 1. There are another two examples of monosaccharides which are also hexoses, such as the fructose and the galactose. Here are the diagram to show the structural formula of the fructose and galactose. So for first year of matriculation program, you just need to focus on glucoses. These two are for reference. The main difference to differentiate from the glucose is fructose, the ring structure of sugar is seem like a pentagon. And galactose, even though they have the similar shape of the ring structure of sugar, but if we focus carefully, the location of the hydroxyl group at the carbon 2, 3, 4, the orientation is different, which is below, above, above. There are three examples of disaccharides, which are maltose, lactose, and sucrose. Disaccharide is the term started with di, di, meaning two. This is why they also known as double sugar, because they consist of the two units of monosaccharides that join by glycoside bond through the process condensation, which means a water molecule is removed. And this is why they give us the formula, which is C12, H22, and O11. Because for the disaccharide in our syllabus is referred to the hexose, monosaccharide yang terdiri daripada 6 carbon, iaitu dengan formula yang kita study di beginning of the chapter. Monosaccharide with the 6 carbon, the formula should be C6H12O6. So if we have two monosaccharide with the 6 carbon, the hexoses, when they combine, if according to the mathematics, it should provide us the formula, which is C12H24 and O12. But this is not the final result can be observed. The reason is the condensation. One H2O is removed. Satu air telah dikeluarkan. This is why you notice the the number of hydrogen atoms is -2 here. It gives us 22, and the O is 11 because one O atom is removed to form a water. So this diagram shows us the simple representation of condensation. So if we have a small molecule of carbohydrate, here we have two units of sugar that already joined by a glycoside bond. Now they are going to join with another monosaccharide, another sugar. So what happens is a H2O will be removed from the process. And at the same time, energy will be absorbed to form a bond, which is called as glycoside bond. So the outcome of the process is they will form a larger molecule. There are three examples of disaccharide, which are maltose, lactose, and sucrose. Maltose consists of the two alpha glucoses, and lactose consists of the alpha glucose and galactose, and sucrose consists of alpha glucose and fructose. Because of the two difference of monosaccharide found in the disaccharide, it can be joined with different types of the glycoside bond. For example, maltose, the two alpha glucoses are joined through alpha 1,4 glycoside bond. And for lactose, the beta 1,4 glycoside bond is formed between the alpha glucose and galactose. And lastly, the alpha glucose and fructose in the sucrose is formed through the alpha 1,2 glycoside bond. In the first year of matriculation program, we just focus on the maltose, which is the example of the disaccharide that consists of two alpha glucoses that join by a alpha 1,4 glycoside bond. This picture shows us the formation and breakdown of maltose. So remember, this is the last learning outcome in 1.2. So first of all, student must be able to draw the units or the monomers of maltose, which are two alpha glucoses. So the picture or the structure of the alpha glucoses must be correctly drawn, and how the water can be removed through the condensation process. So the H2O, which consists of 2H and 1O, is actually removed from the OH group located at carbon 1 for the first monomer, and also the whole hydroxyl group of the OH group at the carbon number 4 for the second monomer. So this is how a water can be removed. A double arrow is shown in this drawing is because the title formation and breakdown. So from the smaller molecules, which are alpha glucose, the monosaccharide, they will remove the water through condensation process. It will form a larger molecule, which is the disaccharide, known as maltose. So here is the full structure of the maltose, which with a labeling of the bond formed between the previous monomers, called as alpha 1,4 glycosidic linkage. The name alpha because the monomer consists of alpha glucoses. 1,4 because the bond is formed through the water removal involve the carbon 1 and the carbon 4. And to balance the equation, we must end the drawing with one water, one H2O. Okay, so vice versa, if the question asks about breakdown, so we have to draw the drawing in another way around. That means how a larger molecule of maltose can be broken down into the smaller subunit.
Saya ingin menunjukkan lukisan berkaitan dengan tugasan formation of maltose. Okay. So maltose adalah disaccharide that consists of two alpha glucoses. So I would like to draw the first alpha glucose, which I always show the O atom, and then followed by the structure, the ring structure of the alpha glucose, which is a hexose, satu monosaccharide yang ada 6 carbon. So after I complete the ring structure of the alpha glucose, I add on the straight line to represent the bond at the C. Okay, what is meant by this structure is the one I jot down with yellow color is the part that you don't need to mention in the exam here. This structure represents that the carbon 1 is located here, followed by carbon 2, 3, 4, 5. Okay, this is the one that you don't need to show in the exam. Okay, now I complete the drawing. So this is the carbon number 6, which always attached with 2 H atom and 1 hydroxyl group. So CH2OH. Okay, now we fill up the functional group of alpha glucose. Alpha means that the hydroxyl group of the carbon 1 is located below the plane of the ring structure. And carbon 2, 3, 4, they have the hydroxyl group. And the hydroxyl group is located according to the one we mentioned in the lecture, which is BAB. Bawah atas bawah atau below above below. So carbon 2 bawah atas bawah. Okay. Make sure that the OH attached to the bond with the O atom. Okay, not the H atom. Okay, so we complete the diagram with the H. Okay, we complete with the H atom. Never forget it. So this is a drawing of 1 alpha glucose. Maltose consists of 2 alpha glucose, so I complete the drawing of the second one. It's better to draw your alpha glucose bigger to have enough space to fill up the atoms or the function group. So I complete with the straight line, represent the bond. I complete the OH group at the C1, C2, C3, C4. And never forget about our carbon number 6, which is summarized in CH2OH. Okay, after that, we complete the structure with the H atom. Okay. So here is the drawing of 2 monomers of the maltose. Okay, now we show how the bond can be formed between these 2 monomers. Okay, to produce maltose, condensation occurs. So we need to show how a water molecules can be removed. So actually it's the removal of H2O between the functional group of C1 and C4 of the neighboring monomers. Okay, so this is one water remove. Okay, and now we have to move on to the next by drawing one arrow to show condensation, because this is a process involve the removal of water. So condensation process with the product, which is the maltose. Because the space is limited, if can, you should draw your maltose at the right hand side, okay, to make it better. So I continue at the bottom. Okay, the maltose drawing I always draw like this. Okay, I complete my drawings repeating with the method I showed just now. Okay, draw the ring structure, add on the straight line. Okay, fill up the OH group, bawah atas bawah, and then the carbon number 6 always represented with CH2OH, and we have the H at the empty ends. And the neighboring also, I repeat my drawings with the complete ring structure of the alpha glucose. Okay, it seems like we repeat the drawings. Okay, so OH below at C1, then bawah atas bawah, then CH2OH. So I complete with the H. Okay, make sure you draw it completely. Okay, you remember just now we say that the 2 alpha glucoses undergo the removal of water. The purpose is to form a bond between these 2 monomers. So how does it happen is, after I draw it one more time, right, then I erase it. Okay, I erase. Oh, sorry, I have erased all. Actually, I have to erase the OH here and only the H at my left hand side, the first monomer. Okay, so what happened is, now we have left over with this structure. We have left over with this structure. We have left one O atom because the H from the first monomer already combined with the HO of the second monomer. So only left out this H. So my drawing here should be a O that have been held by the 2 monomers. And never forget that our straight line at the top, which is still attached with the H. Okay, so how to label the bond? We use a cursor to label it, which is the alpha 1,4 glycosidic linkage. Okay, the better arrow is to be like this. Hold on, I erase this part. This is better. Okay, now we balance the equation with 1 water. Okay, if the question talking about breakdown, okay, if this is another vice versa question, if talk about the breakdown of maltose, not more formation. And now we look at another type of question, breakdown of maltose. So the formula should be started with the larger molecule, which is the disaccharide maltose. So I used to draw the monomer of the maltose. We do it like a practice. So a ring structure of maltose that already have their skeleton, and another one located next to it have the skeleton of the structure, and we complete the drawing with the carbon number 6, which I always forgot about it. And now we complete it with the function group, OH below the carbon 1, and carbon 2, 3, 4 bawah atas bawah, BAB, atau below above below, so BAB, and complete the ring structure with the H atoms. And remember, this is about breakdown. So about breakdown, which is maltose involved. So here we suppose have to make it a shape of the maltose. Okay, a shape of the maltose. So it should be a O between the two monomers, because this is the one we call it as alpha 1,4 glycoside bond. Alpha 1,4 glycoside. Okay, glycoside bond. Okay, because the bond is formed between the C1 and C4. And the process to break down the maltose, we required a water molecule. So we mention add 1 water. Okay, and the formula will be represented by one-way arrow. The process is the opposite with the condensation. We call it as hydrolysis. Sebab dia melibatkan air, hydro, breakdown, so nama dia lysis. So once breakdown, it form the two monomers of the maltose, which call as alpha glucoses. So I draw the ring structure first before I forget. I quickly jot down the carbon number 6. I jot down the OH of the C1, the BAB, the H. Okay, make sure the structure is complete. Okay, make sure the structure is complete. So I put a plus arrow to represent the equation, the balance equation. Okay, so we never forget to name our products here. I mean the molecules, sorry, the molecules. So this is maltose. This is alpha glucose. Alpha glucose. So from a maltose, we have the alpha glucoses. Okay, so therefore, that's all. This is the drawing of today.
In nature, there are 3 examples of polysaccharide, which are starch, glycogen, and cellulose. But for the first year of matriculation program, we are going to focus on cellulose only. What is meant by polysaccharides? So polysaccharides are actually a sugar consists of hundreds to thousands of monosaccharides. They consist of many monomers. So this sugar is a complex sugar. And the molecular formula is represented by this C6H10O5 in bracket N. Depends on how many of the monomers can be found in that complex sugar. The formation is conducted through the process of condensation. That means the many water molecules will be removed to produce a complex sugar. The process is also known as polymerization since they produce a molecule which is polymer. As I said before, there are three examples of polysaccharide, which are starch, glycogen, and the cellulose. What made the main difference between these three polysaccharides are both starch and glycogen consist of alpha glucose as their glucose monomer. That means the basic unit to produce this polymer is the alpha glucose. So there are many of the alpha glucoses join to form the glycogen and also the starch. So what made these two different is the origin of the polysaccharide. Di mana kita boleh jumpa polysaccharide ini? Starch is the polysaccharide can be found in plant. However, glycogen is the polysaccharide can be found in animal cells. But if we move on to the third example, which is the one more focused in our syllabus for first year, is the cellulose. Cellulose is the polysaccharide that consists of beta-glucose as their monomer. So there are a long polysaccharide chain which consists of minimum 300 to 15,000 of beta-glucoses joined through the glycoside bond and arranged in a long chain. And the chain called as the cellulose strands, berbenang cellulose, it will be aligned or arranged parallel to each other. Cara susunan dia kalau kita tengok diagram di sini, dia adalah selari. Iaitu mereka tidak akan bertembung pada mana-mana point. Ini adalah sebab if you observe carefully, the long cellulose strands, they are arranged and stabilized because there are an interaction between the function group. Dia ada daya tarikan yang mengekalkan posisi dia, iaitu hydrogen bond that we have studied in the water chapter also. Okay. If we study the schematic diagram, you may see another difference here between the cellulose that consists of the beta-glucose with the polysaccharide that consists of the alpha-glucose is the location of the glycoside bond. We notice in this example of the long chain of cellulose strand, they consist of 5 units of beta-glucoses here. So you notice that the glycoside bond are located in the pattern up, down, up, down. This is because the chemical bond between the two beta-glucoses are called as beta-1,4 glycosidic linkage. That means the water molecules removed from the OH group located at the carbon number 1 of the first monomer and the carbon number 4 for the second monomer. But to produce the beta-1,4 glycosidic linkage, the orientation of the sugar should be adjusted. So you notice for the first monomer, the beta-glucoses are the one we notice carbon number 1 started at the right hand side and carbon number 2, 3, 4, 5, they arrange in a clockwise. However, if you look at the second sugar, carbon number 1, even though is located at the right hand side, but we notice that now the sugar is turned upside down. You notice the 2, 3, 4 until the carbon number 6, they are arranged in a anti-clockwise orientation already because the carbon number 5 is located below the plane of the ring compared to the sugar number 1. Why they need to be arranged in this way? Because they want to make sure the OH group at the carbon number 4 can be aligned same orientation with the OH group of the carbon number 1 to remove the water. And this will help the second removal that happens between the sugar number 2 and the sugar number 3. You can notice that this alignment helps them to remove the water removal to ensure the OH of 1 and 4 are aligned at the same plane. And theoretically, the cellulose strengths atau berbenang cellulose will be arranged parallel to each other. Dia selari. And as I said just now, the parallel arrangement is because the presence of the hydrogen bond. So the hydrogen bonds make these two strengths won't close to each other. Tidak akan berdekatan antara satu sama lain, satu jarak akan dikekalkan. Okay, maka mereka tersusun selari. So how the hydrogen bond is formed is actually involve the OH group between C3 and C6. So for the first strand, ini berbenang atas, and the second strand, berbenang yang kedua ini, macam mana dua-dua berbenang ini dijarakkan dan susunan dia selalu pastikan selari sebab hydrogen bond akan dibentuk antara the O atom from the carbon number 3 with the H atom of the carbon number 6. So sama juga untuk sugar yang seterusnya. Di sini, the H atom of the carbon number 6, they will form a hydrogen bond with the O of the OH of the carbon number 3. Okay, dan semua akan berlaku sama untuk sugar-sugar yang seterusnya. So what is the main function of cellulose? It's actually the major components of the plant cell wall. Iaitu, kalau kita tengok dinding sel tumbuhan, di dalam sel wall itu, bahan yang paling banyak sebenarnya terdiri daripada cellulose. Dan kepentingannya ialah membekalkan structural support. Dia bagi kekuatan kepada dinding sel tumbuhan tersebut. So kalau kita tengok gambar di sini, manakah satu adalah cellulose? Dan di sini kita nampak ketiga-tiga diagram ini menunjukkan di sini semua adalah polysaccharide. Tetapi macam mana membezakan ialah kita kena faham ya the simplified diagram, makna simplified diagram itu. This symbol represent the location of the carbon number 6. Carbon number 6. So if here is the carbon number 6, we know that beneath it yang kat bawah dia adalah carbon number 5 di sini. So ini carbon number 5. Okay. And the symbol O atom between the monomers of the sugar represent the glycoside bond. Okay, so here got the glycoside bond. And here is another glycoside bond that show us the branching. Other polysaccharide strength they show branching. Iaitu berbenang polysaccharide itu ada cabangan. Di mana sebenarnya di sini dua daripada tiga ini sebenarnya bukan silibus tahun pertama kita. Tetapi saya hanya nak draw your attention. How we want to identify which one is cellulose? Actually, we can notice the location of the carbon number 6. Selain daripada tengok kedudukan glycoside bond. So the answer will be the second strand. Because why? Only the second strand show us that the location of the C6, the carbon number 6, they are located upside down. Okay, the first one up at above the plane of the ring structure and followed by the below one, and then they repeat the pattern. If the carbon number 6 located in this way, that means their glycoside bond also will be located in the up-down pattern. And this only performed by the polysaccharide that consists of the beta-glucose, which is cellulose. Always remember, only cellulose consists of the beta-glucose. Starch and glycogen both are polysaccharide with the alpha-glucoses, and these two are not in the syllabus of the year.